Okay, so what we're going to do here is start looking at some of the basics about eukaryotic gene expression. First, several key points, and then we'll kind of diagram, if you could say, this is also a common test question, by the way, diagram a generic eukaryotic gene, and then we will have what are the functions of these different sequences. Okay, so eukaryotic gene regulation is much more complex. Just again, like I use the analogy, imagine trying to drive down the road and you had two positions on the throttle, either in idle or throttle full over. You might be able to get away with that on lonely country road, maybe not either, but don't try it in Atlanta rush hour traffic. In fact, don't try it in Atlanta rush hour traffic under any circumstances. The term hell on wheels really applies to Atlanta traffic. I've been there, done that, I don't want to do it anymore than I have to. Okay, but anyway, okay, so we look at eukaryotes. We have several key differences. Now, first of all, of course, we have these three specialized polymerases, each for certain types of RNA. We will mention that the promoters are almost always very weak promoters. We have polymerase, the appropriate polymerases. Okay, we have the polymerase, the appropriate polymerase, and a number of what are known as basal or general transcription factor. The term transcription factor refers to a protein involved in transcription. In biochemistry, if you find some kind of biological action, but you have no idea what the hell it is, you call it a factor. And then later on, you characterize it, figure out what it is and what it actually does. But usually the term factor means we've got some kind of molecule that performs some kind of function, but we really don't know what it is yet. That's, please give us a lot of federal money, NIH money, so we can find that out, right? Come up with your team help. I have found a factor. I could identify what it is if you give me a nice big fat juicy NIH grant. Okay. All right, but anyway, the basal or general transcription factors plus the polymerase form this thing called the basal complex. And we're talking about half a dozen additional proteins, many of them multi-subunit proteins. So the basal complex in eukaryotes is humongous. It's very, very large. Okay, so we have that. Another big difference. Okay, another difference. Instead of like a single operator sequence, we have multiple sites. Some of them close to the gene, some of them as much as a couple thousand base pairs away from the gene. These sites are often called enhancers. Sometimes they call them upstream regulatory elements, especially for ones that are a little bit closer to the gene compared to ones that are like within a few hundred bases compared to ones that are a few thousand bases away. The terminology is kind of a little bit fluid and stuff. You'll see both terms referred to. Ones that are predominantly repressing expression are sometimes called silencers, but I think that term's fallen somewhat out of favor. Okay, and what these things do is these sequences bind numerous different transcription factors, especially what we call specific transcription factors. This is the key to cell-specific regulation of transcription. This is one reason, for instance, this is a major reason, for instance, why The globin genes that form the protein components of hemoglobin are expressed and transcribed at very high levels in the erythroblast, the red blood cell precursor. So they're cranking out huge amounts of hemoglobin. All the other cells in your body have the globin genes, but they're rarely transcribed. Occasionally you get some mistakes, and we'll catch that later on. We'll catch those rare errors up later on. But the key thing is, transcriptional regulation is the key, and the erythroblasts have a particular specific transcription factor that ramps up transcription of those globin genes extremely high levels. Other cell types don't. So they don't transcribe the globin genes. Once in a while they make a mistake, but usually not. Okay, so that's an important thing. This is where we get our cell-specific regulation of transcription. And in a higher eukaryote, a typical gene may well have as many as a score of separate sequences that bind regulatory proteins. And those proteins a single enhancer may be able to bind several different proteins. They have to compete with each other for the same binding site. And we'll see all these transcription factors, or almost all of them at least, are regulated. So they can be turned on or turned off by things like phosphorylation, binding to other proteins, any of a number of things. They are regulated proteins, and once again, I cannot overemphasize the importance of protein regulation. Trust me, you know that the hard way. Okay, but regulated proteins. Okay, now, many of these transcription factors are effectively what we would call activators. In other words, they tend to ramp up transcription. But there are others that are repressors. They tend to inhibit transcription. Okay, and sometimes the same protein can act as either. So a given protein may be an activator, may increase transcription in one situation, and then you phosphorylate it or bind another protein to it or whatever, and it changes tune and becomes a strong inhibitor of transcription. So sometimes the same protein, some proteins are mostly activators, some are mostly effectively repressors, but sometimes the same protein can do one or the other depending on how it's regulated. So that throws an additional complexity to this whole kind of thing. That's why we can get development in the first place. You don't see multicellular bacteria around. Yeah, they cling together, they communicate with each other and stuff, but they don't form a true multicellular organism. But eukaryotes, at least, especially the higher eukaryotes, can. And that raises up the bar for the complexity of transcriptional regulation. So we have that kind of thing. We have these multiple sites, lots of places to control, things to compete with each other. Some will lead to activation of a gene, some will repress the gene, they interact with each other, the proteins interact with each other, and you get an extremely complex network of interactions that people are just beginning to try to investigate. So it's a very complex thing for every single one of our genes. Okay, so you have that. Now another thing, this is also what we find in eukaryotes. I think most of you probably know this. Now this is something that we do not find in prokaryotes. But in the vast majority, there are a few exceptions. I'll point some of those out when they come. The vast majority of eukaryotic genes are split into different regions. We'll have, here's the gene here. And we will have a region, we'll call it an exon. That region contains the instructions for making a portion of the protein. And then you have interspersed these regions that do not have those instructions, what we call introns. And then we have an exon, and then another intron, and let's say another exon. Okay. Often, by the way, for future use, I just did this because sometimes people think that these are completely separate pieces of DNA. They're not. Okay, so I'll put exon. This is usually my conventional way of doing it. Okay, so this is just, we'll be seeing this a little bit more often. Okay, this is still a continuous sequence of DNA. This entire thing still is the gene, but here's the exons and the introns. Okay, now a good analogy for that is watching a film. Go out and buy a DVD, or rent one, or whatever, Netflix, whatever the case is. Now, once the film starts, you get rid of all the trash and garbage at the beginning, and the FBI warning that will throw you in jail if you copy it, even if you're not making money off of it. And but anyway, once you start the film, it's a continuous storyline. If you have to go and take a pee or something like that, or you want to cook some popcorn or something, if you don't pause the player, you're going to miss part of the story. Okay, that's like what we see in prokaryotes, a continuous unbroken coding sequence. Now, in eukaryotes, it's like seeing the same film on network TV. What do you get? You get a sequence of storyline for 10 or 15 minutes, and then you get 5 minutes of commercials that have absolutely nothing to do with the storyline, and that's when you get up and take a pee or cook your popcorn or whatever the case is. So, and then you get another segment, another film exon that has the storyline, and then another battery of commercials, another intron, and so on and so forth. That's why a 90-minute film runs to 2 hours, even with the censors cutting parts out on network TV, because it's so full of commercial introns. And of course, the introns have nothing to do with the coding instructions of the protein. Now, sometimes it will have regulatory sequences in them. Sometimes introns have genes in them. That happens. But it's not direct. Generally, they are not direct. They are not part of the coding sequence. And just like commercials, you see commercials on a film or something, half the time they have absolutely no relation to the storyline of the film. And sometimes they have something really that is not only completely off, but also embarrassing, insulting, or whatever. I'll give you one example that's happened many years ago. They had this, award-winning BBC series on World War II for like about 15 or 20 episodes or something like that. And they had, not surprisingly, a full, segment on the Holocaust. And what do you know? What did they advertise in one of the commercials? An ad for oven cleaner. I saw that, I'm thinking, great, do you have a dirty oven? Yeah, I just murdered 6 million people and it's really a mess. Needless to say, Easy Off got a whole bunch of rotten egg on their face for that one. So they have to be a little more careful about commercial placement. That was one of the worst ones. I heard some other pretty bad ones too, but sometimes you have to watch what you're placing your commercials. But that was completely different than the topic of the thing. So that's what introns are. Now you may wonder, why do we do this in eukaryote? Doesn't that take chances that we're going to accidentally, in a final messenger RNA, lose parts of exons, or throw in parts of introns we don't want? Yes, there is a risk of that. But the key reason for doing this is it gives us additional, through RNA processing, gives us additional levels of control. Sometimes, what we call alternate splicing, we could do things like, we'll take exon 1 and 3, but get rid of exon 2 in one cell type. Another cell type will have all 3 exons, and a third cell type will only have exons 1 and 2. You get 3 different messenger RNAs in 3 different cell types. Means the same gene can code, in this case, for 3 proteins, 3 distinctly different proteins. They may have similar functions, but they will be cell-specific versions. That's one thing. And there are some proteins that are coded that actually have, I think the record breaker has over 300 exons and introns. It's estimated that it can code for something like 30,000 different cell-specific versions. And each cell type has, and it changes during development too, has a particular version of this. And this kind of protein allows cells to recognize and stick to similar cell types. So, in other words, you can have one gene coding for a whole battery of a whole family of related proteins. So that way, that's one reason why we have only about 22,000 genes. This little tiny millimeter long hair width thickness roundworm has 18,500 genes. We can make a lot more different proteins off the same gene, and those guys can't. We're a lot more complex than most of us, I think, at least code. Well, I don't know, some of the freshman folks are sometimes I question that, you know. But at any rate, for the most part, you know, we're a much more complex organism. We can actually get by with fewer than hundreds of thousands of individual genes would actually need. If we had every single protein coded for by its own gene, we would fill up our genome to the gills and then some. So that would be bad. That means high mutation rates and nasty things like that. So actually that's a good idea. A second thing too is when you get, for having things separate, is you can get these things like duplications and translocations of DNA segments. So let's suppose we take this exon, we copy it, cut and paste. And we happen to paste it in front of or somewhere, maybe even inside another gene. Okay, so let's cut and paste, and we'll paste the copy, or rather copy and paste. We paste the copy inside another pre-existing gene, and what do we have? We have the original gene with this extra segment in it. Now, most of the time, those kinds of translocations are going to be damaging to the gene. It won't function properly. But sometimes what you can do is you can add in a new function, or maybe a new way of regulating things. A lot of times, the exons code, this is not always the case, but a lot of times exons code for functionally distinct regions of a protein, like a regulatory site, or sometimes you'll have one or two exons will contain the active site of an enzyme, sometimes only in one exon. So what you have in effect is a modular protein. You can take a module from one protein through duplication and translocation, stick it onto another protein, you may get a protein, a modified version of that protein that has new capabilities and new functions. It's widely believed that most of our complex proteins, the ones that have molecular weights in, you know, several tens to a few hundred thousand stuff, many of them, if you look at the protein, the gene sequences, it looks like they are glued together. You find sequences that are very similar to this protein, a part of that, and sequences very similar to a different kind of protein somewhere else, and so on. So it looks like a lot of our complex proteins are glued together through evolution by smaller modules. This will increase that process. It can separate out chunks, and most of the time the introns are far longer than the exon. I mean, I make the introns short here, but in real life, introns are often many times larger than the given exons. Okay, so this allows us to do this cut and paste function that generates novel proteins. Most of the time it screws things up, but sometimes you get something that's novel and plays a function. So a lot of times you look at things like protein sequence, and I refer to things like immunoglobulin loops. In other words, these seem to have been derived from the antibody or immunoglobulin protein. And found a whole bunch of things that have nothing to do with your immune system. And something called an SH2 region. I think those who remember cell biology, we mentioned this SH2 family protein. It's named from a particular structural region found in a protein called SARC. And a whole bunch of other proteins have that region in there. And there are numerous kinds of things. So this also allows the evolution and development of more complex proteins that can do multiple functions, that may have multiple ways of regulation, things like that. So these are actually a good thing, and for the most part, it's worth the risk that you're not going to splice the exon sequences together probably. Usually I don't make too many mistakes, but it's worth the risk. Okay, so that's something that is unique. The introns and exons are unique to the eukaryotes. And it gives us a lot more diversity. One gene could potentially code for a broad, diverse family of generally similar proteins that have similar functions. But sometimes the different proteins actually do have distinctly different functions. So that's another kind of thing. Now, and we're going to actually, after I draw a gene sequence, we will enter into this. DNA, now if you look at bacterial DNA, you have this big one or more big loops of DNA, may have some plasmids in there, all coiled up in the center of the bacterial cell. For the most part, there are proteins associated with the DNA, but it's not really coded in a huge amount of protein. On the other hand, eukaryotes, that's a totally different story. Eukaryotic DNA is not naked DNA. It's complex to a whole bunch of proteins organized in a very careful, specific fashion to make what we call chromatin. And during mitosis, this packs up real tightly to form what we call chromosomes. Okay, so now, when we were looking last week at those interphase chromosomes, you can see kind of this model appearance in the nucleus compared to the ones undergoing mitosis or meiosis. And that was the chromatin. You couldn't figure out how it was arranged and stuff inside the nucleus. It was splayed out, but you can see picking up the stain and gave the whole nucleus a model appearance. When you look at the ones that actually had meiotic chromosomes, and then they appeared as little tiny, very dark dots. Remember those things? No, not, you might need to, very shortly. Okay, all right, so we have chromatin. Now, one of the major features of chromatin, one of the most important parts of organizing this, are a set of proteins known as the histones, which associate with DNA to form structures called nucleosomes. That's a core structure of chromatin. Okay? A complex of histone proteins plus DNA. We're going to find, and we're realizing more and more the vast importance of it. At first they thought this was just a way of packing 2 meters of DNA. The total DNA in our cells stretched out, and of course that's at 46 chromosomes, but stretched out, laid out end to end in the nucleus of one single cell. We're talking about something that's, a couple to maybe 5 micrometers across, so small you need a microscope to see it. Okay, well, pack 2 meters of DNA in. I don't think anybody in this class is 2 meters tall. No, you're not quite close. Almost. Hello? Yeah, text your friends later. Yeah, not good. Lose stuff. Okay, but anyway, okay, so this is the fundamental packaging material to organize that much DNA into that small space without getting everything hopelessly tangled and causing all kinds of problems. So first they thought, okay, nucleosomes and all this chromatin stuff is largely for packaging DNA. Of course, it is very important, but now we're realizing that especially the nucleosomes are very important in regulating gene expression. Because what they do, nucleosomes, the histones bind so tightly to DNA that nothing else can displace them, or almost nothing else. So, nucleosomes are repressors of transcription. The default situation in eukaryotic genes is the genes are off because key sequences like promoters and stuff are tightly bound to those tightly bound to the histones in the form of nucleosomes. They're bound so tightly that all the other proteins, the polymerases and the basal factor, transcription factor, just can't get to them. So that means genes off. So in other words, it also says when we want transcription to occur, we have to do something to those nucleosomes. We have to weaken the grip on DNA, get rid of them, push them to the side, or whatever the case is. And we will find that that is absolutely critical for transcription to occur. You have to do something to those histones. You have to regulate them so they don't bind the DNAs tightly, and then you can slide the nucleosome away from key sites, push it off, whatever the case may be. That's essential for transcription to even think of occurring. So, we're finding this is very important. Regulating these histones by multiple means is very important. And sometimes we can regulate them long-term, make changes that are very hard to undo, and nearly permanently inactivate a gene. If we're pretty sure, it's like getting, what we could call genetic vasectomy, right? You don't want more kids anymore, you don't want any transcription anymore, you go up and get snipped. Okay. You're really sure you don't want, if you're really sure you don't want to transcribe that gene anymore, you can modify those histones in such a way that that gene's pretty much permanently shut off, and it takes a great deal of effort to undo that. That's sometimes what we call epigenetics as part of it. We'll see that. A lot of these long-term epigenetic changes, some of them which can be passed down from one generation to the next, those frequently involve stable modifications to the histones. And they can either strongly and long-term repress transcription, or in some cases, they can tend to activate transcription, make its activation much easier. So that's an important thing. Okay, so chromatin and the organization of DNA, it's not just packaging material anymore, it's critically important in short and long-term regulation of transcription eukaryote. We don't see that in bacteria. Now it turns out, although it's a far simpler system, it turns out that archaeas, the bacterial-like organisms, are a third domain of life and probably the closest to the last, what they call the last universal common ancestor, Luca. But anyway, it's likely that both eukaryotes and bacteria evolved from archaean ancestors. Okay. But anyway, they actually do have histones, and they're broadly similar to ones we have, but their regulation is quite a bit different. Okay. All right. Well, that wasn't too long. Ooh, all right.